Welcome, everybody. Thank you for coming out tonight and joining us in the dark. Thank you for your courage to leave the house in the rain. I'm appreciating everyone's presence already with us. It was nice to have some chatty vibes in the space. My name is Hannah. For those of you who haven't yet met me, I am now the guest interviewer for District216. Thank you to Jacob for this opportunity. Is anyone here brave to raise a hand if it's their first time at the district house or at a district event? Cool. So we all have a sense of familiarity with the space. But just to recap, we're here to present in a way that is entertaining and educational around art, psychedelics, cannabis, music, all the things that Jacob loves, right? So tonight we have, I love that you said this about yourself, Connor. If Alan Watts was a neuroscientist, he's in the building tonight representing his research from UCLA. And I'm appreciating your backstory, what you're working on, and we're going to delve deeper into those details. But if you would like to give a brief intro of who you are and perhaps why you're here.
Sure, yeah. Thanks so much for having me here, everybody, and excited to present some data from the work I've been doing in psychedelics. And just to kind of contextualize what I was saying about Alan Watts, the whole reason why I got into this work in the first place is because I had some personal experiences, like a lot of folks do, related to meditative states, let's say, and was curious whether or not these had been reported by other people. Mine were induced partly by cannabis, and so I was researching this on the internet and found an article by Alan Watts called Psychedelics and Religious Experience, and I just thought he was an internet blogger at the time and had no idea who Alan Watts was, but it was so powerful. It was making me cry actually reading this because I felt that I could have written what he was writing, describing these experiences, and I realized this is not just something happening with me, this is something that can be reproduced in another person. And so it became apparent that trying to study this scientifically was going to be an important thing in the future, and that's kind of what I've been doing since then, is bringing this into the neuroscientific community as much as possible. So trying to live up to what he would do if he were to go and get a PhD in neuroscience, and then the questions he might be asking is what my kind of career motivation is all about. Yeah.
It's super cool, right? And I must share, I had an assumption that your awakening was catalyzed by LSD because that's what your current research is oriented around. And so when you shared that it was more so cannabis and finding Alan Watts, I thought that that was super curious and cool. And then you spoke to actually being invited into that topic of research. So would you like to further share the questions that you are currently exploring in this research?
Yeah, absolutely. So, when you want to pursue a career in this, you realize you have to go to graduate school and well, what do you study? And so, I found studying at the time the neurobiology of substances in the context of addiction was one great stepping stone to get me to the next level, which ended up being a human research lab where they were studying drug effects in people. And specifically, I wanted to study cannabis and these types of altered states. And the professor, Harriet de Wit, said, well, that's great. You want to study cannabis and altered states, whatever you're talking about. But we actually have a microdosing LSD study happening now. So I'd like for you to work on that. We have an MDMA study happening now. I'd like for you to work on that. And I was like, this is really exciting. I mean, all the things that I am interested in with cannabis are within this larger context of psychedelic effects. So to be able to get experience working with microdoses of LSD, even though I had no idea at the time what the hypothesis was or anything she was looking at with that, it was super exciting to be able to get into that world. And now being able to apply my own analyses, being able to compare effects from THC, which is the major psychoactive component of cannabis, to effects of microdoses of LSD to other drug conditions allows us to have a really good sense of what these different compounds are doing in the brain and what they might be able to provide in terms of clinical effects. So I was able to, have much more than I bargained for by joining her lab in Chicago, and that's where all the data that will be coming from my talk today is originating from.
Super cool. And I am appreciating learning from you already because of the very linear and objective approach you have to this research. And I'd love for you to provide your own clinical definition of psychedelic effect and what that means to you.
Yeah, psychedelic is always a fun question. What is a psychedelic? And the way I've been thinking about it recently is it's a noun and an adjective. So in the noun sense, you can think of it in terms of like the classic psychedelic, like a 5-HT2A receptor agonist. So those things like psilocybin or LSD or DMT acting on that particular receptor, it's going to mediate these certain types of effects. And the adjective version is the psychedelic effects, where the psychedelic word came from in the first place. This mind manifesting type of effect might not be limited to a 5-HT2A agonist, might also be from a cannabinoid drug or from just meditation without any drug at all. And so thinking about psychedelic in terms of an adjective, that's more my research interest. And thinking about psychedelics as a noun is helpful, I think, from a chemistry or pharmacology standpoint.
Yeah, and I like what you shared with me earlier as well about that and the term hallucinogen. So do you want to present the relationship between those two things?
Yeah, so they're used interchangeably, hallucinogen and psychedelic. And I actually have a mentor of mine who prefers the term hallucinogen because of hallucinare, which means a wandering mind. But if you look at it, mind manifesting means that there's something that you're coming into realization or insight into. You have something that you notice. Maybe it's about your mind. Maybe there's some self-awareness aspect. Whereas hallucinogen is like a delusion kind of thing happening. And so to me, they're more antonyms than synonyms. But I think they also can come in the same drug effect sort of array of effects that might happen. So for instance, you might take drug A and have some psychedelic effects, but you might also have some hallucinogenic effects at the same time. Like LSD, for instance, you might have some mind manifesting effects, but the walls might be melting in the background, which would be more of the hallucinogenic effect. Whereas drug B, you might have more psychedelic effects and only a minor amount of hallucinogenic effects. And those are some questions I'd like to pursue in the future. How much, what's the highest ratio of psychedelic to hallucinogenic effects that we can get from a certain compound? And maybe meditation, for instance, is 100% psychedelic and has no hallucinogenic component. So those are things I'm interested in as well. Or maybe there's some hallucinations happening in meditations as well. So yeah, I think those are all really worthy areas to investigate.
Yeah, for sure. And I just have a personal curiosity. Is meditation something that you have explored in your personal practice?
It goes back to those experiences I had with cannabis, which it turns out adolescents actually have more cannabinoid receptors in the brain. And so your brain's kind of more in tune to receive these types of experiences, I believe, in adolescence. And I did a study, actually, and I'll show some data related to it, where indeed people who are in adolescence, so like 18 to 20 years old, will have stronger effects in terms of cognition and behavior from cannabis relative to the age I am now, which is between 30 to 40, was that older adult group. But in terms of being younger at that time, having those types of psychedelic experiences. Actually, I'm sorry, can you rephrase the question or repeat rather the question one more time?
Yeah, I was asking if you have a personal meditation practice.
Oh, yeah. So what I found was that it was inducing meditation. And so I like to kind of use the word meditative agent sometimes. You'll take the substance and it is, we use the term therapeutic agent often in clinical pharmacology, but meditative agents is something that are inducing this state without you really needing to think about it. And so I don't have a particular meditative practice, but certainly my background comes from these types of meditative states that were induced, I believe, organically. It was sort of unexpected. And it's kind of, it goes back to those stories you hear with scientific discoveries being made on accident. I wasn't trying to have a meditative experience from cannabis. I was just a high school student, experimenting like a lot of other high school students were, but stumbled upon this type of effect, which I felt was going to be important to pursue.
Yeah, and I want to acknowledge how amazing it is to meet someone who's so on purpose, and that you had these early experiences in high school and that defined your professional career in a way that was personally impactful and now is being so impactful to the community and beyond. And I think that's really special that you found that, or it
thanks.
found you rather.
Yeah, thank you.
Yeah. So I know that the University of Chicago is where you were initially introduced to this research project. And you shared with me a super curious story. And would you like to share that with the audience now?
Yeah, sure. So I was only in Chicago for 2 years, but the most interesting day that we had was during our MDMA study. So this is a study, the design was to look at folks with autism. And the hypothesis was that folks with autism wouldn't like to be touched by people as much as a healthy control. And so what we did is we actually had a paintbrush and we would brush their arm. It's called an effective touch. The paintbrush feels really good, and it might feel even better if we give you MDMA. And if you're someone who has autism, you might feel so good that now you feel like a healthy control person when we're brushing your arm. And the great thing about the studies that were done at the University of Chicago is that they're very well controlled in terms of what we call expectancy effects, which means if I have a study with MDMA and I'm telling you, you're going to get MDMA, you're going to have this expectation that you might have those effects you associate or hear about with MDMA, like a lovingness type of effect. However, these studies are designed such that there's a whole list of potential compounds you might get in this study, and you have no idea which one it might be. And also to qualify, you could have just had one prior use. So this particular individual on this particular day had MDMA years ago, had no idea he was going to get MDMA. But at the end of the day, after his MDMA session, after receiving this effective touch, said that on his final questionnaire, he wrote down, "I know what I need to do now. Just Google me." And it was kind of alarming to the research staff. What does this mean? Did we just induce some response that he's going to go do something violent? That was kind of a concern. He was giving off kind of vibes that, we weren't quite sure what was going on with this guy. So we followed up, and it turned out he was a white supremacist and had been traveling to Poland for these white supremacy conferences. And basically what he said was that he experienced love for the first time. And that was recently published by the BBC, reported on that. So this was an interesting case that came out of this particular study, and we were not trying to see if MDMA can change white supremacists. That wasn't the point of the study, but it was a really interesting kind of case that came out of our hope that maybe MDMA could improve responses to being, touched by another person. And so in some sense, we did do that, I think. Yeah.
He was touched by love.
Right,
in a way that he hadn't experienced before. Mm-hmm. Yeah, that's so cool. Thank you for sharing that. And after your transition from University of Chicago, you've landed in UCLA, and you recently received some exciting funding for some new projects.
Yeah.
Will you share about that?
Sure, yeah. So the data I'll talk about today were microdoses of LSD, but now at UCLA, we're giving what you can call maybe a macrodose, 25 milligrams of psilocybin. And this is what you would basically see at Johns Hopkins, these studies where you have psilocybin-assisted psychotherapy. So it's a whole 6 to 8 hours. In our particular case, we're going to look at folks who have cocaine addiction. That's kind of my background from the schooling that I did in graduate school. And we're going to look at addiction in the brain, see if it can be reversed from the psilocybin treatment. But first, we got to see if it's safe in this population. So it's a small pilot study, 10 people with cocaine use disorder will come in, we'll give them the psilocybin doses and see how that works for them, and follow it up with some neuroimaging studies later. That's the idea. Mm-hmm.
Very cool. I just want to reiterate my celebration of you moving forward, like doing the research you want to see to impact the world. And it's just really awesome, like the speed that this is all happening. It's all so new and so much has been discovered. And so it's my sense there's much more to discover. Mm-hmm. And you have this project at UCLA that's budding. And also you mentioned to me this citizen project that takes a little bit different shape.
Right, yeah. So the data I'll be showing today from University of Chicago, it took, 2 to 3 years to collect basically 60 people's worth of data, where an alternative model is what they call citizen science, where as opposed to having participants come to the laboratory, you kind of bring the laboratory to you guys at home. And so what we can do now is we can leverage what they refer to as mobile neuroscience technologies like mobile EEG headbands, where we can send you a headband in the mail and you can actually receive reports back at the end of your participation as well. And so that's something that we see in the laboratory too. People want to get their data insights back. Like, can I see a picture of my brain? And usually in the laboratory, we're like, no, that's not what this is about. Thanks for your data. We paid you. Get on your way. With citizen science, you can kind of put it more in terms of putting the participant first. They're actually willing to purchase these neuroscience devices. They'll have them at home. They'll get to keep them, and then they'll send us their data, and then we analyze it for them, send the data back, kind of like a 23andMe or Ancestry.com, but it's for the mind and the brain as opposed to DNA. And we're also opening it up, so it's all open science on Open Science Foundation, so any researcher could access the data and run their own analyses. So this is an emerging area, the citizen science idea, and so I'm now working with a team of people to do this citizen neuroscience, to have people track their brains and see what happens like after a microdosing protocol. And so we just got some interesting results from that last week, which I can kind of answer in a Q&A later about what we found there.
It seems limitless now, this study of psychedelics and the brain, and to be able to do that in your own home is just something that I would have never thought within myself until you shared about it. So I, again, am celebrating these projects and these pathways. And when you consider what is unfolding with both the citizen science and the UCLA project, what else are you looking to research beyond the current cocaine-addicted brain treatment with psilocybin and LSD?
What else? Was there something in particular we were discussing earlier that, or is this just a new kind of an open question?
No, not necessarily. I'm just sort of offering if someone was like, hey, I have unlimited funding for you to use in the way that you want to. What do you want it for? Yeah,
it would go back to the Alan Watts kind of investigation. So the paper of psychedelics and religious experience, he talks about psychedelic awareness a lot. And that idea of psychedelic awareness isn't really on anyone's minds in academia right now. But this idea that we can leverage these types of compounds to potentially access greater insights and observations and self-awareness, I think it's it goes outside of the clinical model. So right now, all of the psychedelics are in the psychiatry department to treat mental disorders and things like that. But ultimately, I think that their biggest power and effect is more of a basic science thing as opposed to an applied science thing. So what I mean by that is just the basic fundamental nature of mind or of being or of reality, those kinds of questions. And it gets really tricky in terms of trying to figure out, again, what's a psychedelic effect and what's a hallucination. So if I could have any, large project, it would be pursuing the psychedelic awareness question, having a lot of people go into altered states and be able to report back on them. But I want to have a caveat here, especially with the citizen science, is that it gets tricky in terms of not wanting, keeping the participants' health as number one. So fortunately, within a clinical environment like UCLA, there's a lot of controls and safety protections in place. For instance, the psilocybin session, we're going to have two MDs, two psychiatrists sitting there in the room, making sure everything's going to be fine and that these people are always going to be watched. The citizen science gets a little tricky when you're wanting people with microdosing. It's okay. You can think, they're probably going to be okay. But, I'm moving into what would be like a ketamine clinic study, and we would want to have controls, making sure that these people are under supervision, that they're in a clinic that they can be cared after. So even though I want to collect a lot of data, I still want to make sure to kind of have that Hippocratic oath of do no harm and make sure people are safe. I'm interested in what you can call psychonautics, sort of like an astronaut for the mind, but there are dangers associated with traveling up in space as an astronaut, as there might be traveling into your mind too. So I want to be really careful with all of the health related implications, even though to me those non-health types of effects, those just basic awareness questions to me are the most interesting and important, and ultimately could help the health of the people and the health of the species. And so I'll just kind of end this answer on this point, which is that Charlie Grobe, who's one of our mentors down at UCLA, who's bringing the psilocybin studies up to the standard that they need to be at. He's one of the first people who did a clinical trial with psilocybin in the new sort of era with publications in 2011, end-of-life cancer patients. He's got really great reports of folks who've kind of gone through those studies. But one thing he'll commonly go back to is this effect in terms of our species' ability to reconcile our sort of existential crisis we face in his mind in terms of environmental aspects. But I think about it also in terms of just our human-to-human interactions in terms of warfare and things like that. And so if you can think of these experiences as you're walking away and having a greater peace, whether it's to the planet or to each other or to yourself, to me, that goes beyond, that transcends medicine, and it's much more of like an evolutionarily driven thing that matters in terms of the species in our future more so than just treating someone's depression. But obviously, that's really important to do too. Yeah.
I received that. I appreciate just how you speak and how I am resonating with what you speak, because I come from a totally different world that's leaning more towards like spiritual, like foo-foo talk. And I'm really just grateful to feel your groundedness and where those two worlds are actually connecting in your vision of this research as it continues. And I would like to shift into our change your lens rapid fire questions.
All right.
But I have a desire to customize them a little bit based off of what I know about you. And so it's my sense that Alan Watts has been a huge influence on your path. And I'd love for you to share something that you've read or heard of Alan Watts that particularly struck you and has really impacted you in a positive way.
His characteristic he describes in that paper I was referring to of polarity is one that really struck me intensely. And you can kind of think about that as like the yin yang sort of phenomenon. But when you experience something like that for the first time without having someone else describe it to you, I think it's really powerful, and it's in some sense like ineffable. But Alan Watts was able to put that into words. So that was one of the things that really struck me from his paper, and I'm continued interested in pursuing scientifically.
And for those who may not be familiar with polarity or yin yang, can you give a little recap of just your own definition of what that is to you?
Sure. I think when he was describing it, and the way I would describe it too, is that things that are usually thought of as opposites can also be interdependent. And so there's sort of this accordance between light and dark, or any sort of in the fact that these polarities exist all over nature, and being able to see those types of polarities in yourself, and then finding that accordance between the two opposing sides into this harmonious thing. That's something that I think he describes a lot, and again was something that resonated with me. And to me, there's a structure there, there's an architecture there, and there's mechanisms there, and there's mechanisms in the mind, and there's mechanisms in this reality or being, and so those are scientific questions, and I think that we can access those through this type of research.
Awesome, thank you for that. And another question I'd like to ask you is if you can recall the details of a profound experience you've had in an altered state. And maybe that was a cannabis-supported meditation or another experience you'd like to describe.
Yeah, this is a funny topic because we go back to that idea of expectancy effects. And so I am... this idea of having a database of altered state experiences is something I want to do, but I'd like to build it in such a way that you can't access certain parts of the database until you contribute to that part of the database. So if you had some type of yin-yang experience and you report it, then now you get to read all the yin-yang experiences. And so now you're not going to have that influence of other people. And so I get kind of, I don't want to go into the full detail of all of the types of experiences that I had, because I want people to independently have those types of experiences on their own. But to give one that is a little more, I guess, mundane, trying to decide, well, I have a bunch of notebooks at home in my closet. So when I was in high school, I would kind of journal in these altered states with cannabis and sit down. And this happened because the first time I had one of these experiences, I was like, I need to write this down. I need to get a pen and paper. And I'm sure people might have had that type of experience too. And I think Carl Sagan also, there's like an article he wrote under a pseudonym Mr. X, where he's talking about some of the most important work he's ever done was to try to write down and get recorded some of these experiences he was having, because they're so fleeting, especially in the cannabis state. You're sort of in one moment and then the next, and then you lose it. And so the ability to record these, I think, is an important methodological consideration. But let me try to find something that I can think back to. Well, I'll just say this. This isn't too crazy in terms of, what people have probably experienced before. You might resonate with this. So this goes back to the polarity idea and with geometry. And I think geometry is really interesting, talking about kind of the objective structural nature of reality. And sort of this realization when you think about 2 points as being opposing one another, but then the accordance between them or synthesis between them is a new dimension. It's a line that connects them. And then again, you have another line, and these two lines connect in a plane. And so each time those polarities combine, there's this elevation and dimensionality, and kind of feeling that through ultimately into time and space, with moments shifting, and then ultimately in terms of you as this entity that has this space-time experience, and then someone else having a space-time experience, and then that higher dimension of our connection with one another as being this reality that exists, and kind of experiencing that in these meditations was, I think, something that other people might have tapped into, but I've definitely written about that a little bit.
I can connect with that geometry interest, and there's a field of study called biogeometry that I would love for you to look into on your own time if you haven't yet heard of that. Super interesting stuff.
Thanks.
Yeah. So I understand you don't have a super passionate relationship to LSD personally, but I do wonder, is there any substance that you're fascinated with but haven't yet tried but hope to one day?
I think, ayahuasca seems interesting, I guess. I really don't know. I haven't really thought too much. I kind of moved. One thing I've described myself before as well as like a psychonaut turned neuroscientist. So I feel like the psychonaut phase is in the past a little bit for me. I'm interested in studying other people who have these types of experiences. So I haven't really thought too much about, revisiting that type of journey in myself. So I guess the short answer is no. Yeah.
You shared a little bit about the MDMA study and that individual's experience of awakening to love. And what we haven't talked about is your personal spiritualism. And that's not necessarily where I'm leading you, but I do have one question. And that is, do you personally believe in an afterlife? And do you think that these substances give us glimpses of what that is?
Sure. Yeah, I'll say yes to that in both cases. And I like the dissociation example and the dissolving analogy and kind of giving yourself up to this whole. And I think that makes total sense in terms of energetics. And I think there's scientific ways of looking at it, but obviously it's a big mystery. So that's kind of as far as I can take it for now.
For sure. Thanks for that. And my final question for you is, in the most succinct expression possible, can you please describe the nature of consciousness?
Complexity, and that's what I'll talk about today. Yeah.
Yeah, I appreciate that response. Thank you, Conor. Is there anything else that you would like to share with the audience before we transition to your presentation?
No, this has been amazing. I hope you guys have enjoyed the little questions. I've had a lot of fun answering them. So yeah, thanks so much as well for asking.
Yeah, you're welcome. Thank you. Thank you, everybody.
All right. Okay. So you guys want to talk about some data?
Yeah.
We get scientific up in here? Yeah. All right. I'm going to teach you guys some things about neuroscience. So you're going to walk away and you're going to, be little PhDs in a sense. But then you'll also get to know about how psychedelics and specifically microdoses of LSD are going to be acting on the brain. So you can kind of brag to your friends about what's happening with them. All right, so yeah, exploring consciousness. This is going to also talk about these, what we call biomarkers, these markers of consciousness. Why do we need a marker for consciousness? Well, if you're going under anesthesia or if you're in a coma, we need to know, are you really out or not? We can't really ask you that. So we look at the brain and we look at the brain's signals, and that's what we're going to be looking at with these data. But now we're going to look at it with people going under a microdose experience as opposed to under anesthesia or coma, because you can also not only have less consciousness, you can also have more consciousness maybe. This is kind of this somewhat controversial idea or interpretation of the data that I'll talk about today. Okay, first we'll talk about what is a microdose. I'm sure many of you know, but it's typically a 5 to 10% of a typical dose. So in the case of psilocybin mushrooms, this would be 0.1 to 0.4 grams or 10 micrograms of LSD. I'm showing these two articles here from National Geographic and the New York Times because it was really exciting to be a young researcher out of grad school and then have the National Geographic call you up and say they want to feature your work. And, it's part of other studies that are going on, but then having, in a little interview and telling them what the data are about and then seeing it published, both, in these really high visibility, websites and magazines is pretty cool. So there was a lot of questions, though. You can see it's like, is it really working? There's a lot of reports that are out there. What do the science say? What do the scientists, what are they finding? Let's start with the reports. So we were talking a little bit earlier today about James Fadiman. James Fadiman is well known for collecting a lot of microdosing reports. He's actually in the kind of lore and mythology of psychedelic research. He was at Stanford in the 60s. He was doing problem-solving studies where LSD was given to folks who had a kind of blockade in their ability to solve problems in their careers, and he was seeing if LSD would facilitate their solutions. And then he got a letter in the mail from the government that said, you got to stop this research now. And he was like, well, I'll just pretend I got this letter tomorrow so these people can finish their session. Unfortunately, it kind of stopped his career in psychedelic research, so he sort of went a little more underground, you could say, and then ultimately wound up being very interested, and now is kind of considered the godfather of microdosing. You might have heard of the Fadiman protocol, which would be taking one dose one day and 2 days off. So anyway, he receives all these claims, and here's one of them in terms of what people are reporting: feeling more love for themselves, cravings going away, their personal and spiritual growth is being affected. So almost anything under the sun. He likes to point out this particular slide. If you look, just going from top to bottom, somewhere in there is a no post-Burning Man crash. He likes to point that one out. So other researchers would say, for instance, Harriet de Wit, where I was working with her back in Chicago, she would say, well, if it's helping everything, then maybe it isn't really helping anything. Maybe this is something where we need to be cautious and try to figure out what it actually might be doing, or maybe this is just a broad stroke kind of placebo effect. That's where the placebo-controlled clinical trials come in and help us get really interesting data. So this big question mark of, are the anecdotal reports true? Do they live up to what we might find in a rigorous placebo-controlled study? And James Fadiman will tell you, these are still data. This is still evidence. It's just a different level of evidence than what we would see in a laboratory environment with a placebo control. So let's get a little bit more rigorous here. Let's get a little more stringent in our analyses. The first thing you might want to look at when you're doing a study of microdoses is whether or not it's actually getting into the body at all, and tracking the amount of LSD, for instance, in this case, in the body over time. So on the bottom of this graph, what we call the x-axis is time. This is just during a session. You can see it's a 6-hour period, and you see this nice increase and then ultimately fall of LSD in the blood. So they're just looking at the blood and looking at the number of the quantity of LSD at these different doses, 5, 10, and 20 micrograms. So yes, okay, take a microdose, it gets into your body, goes up, goes down, that's great. What else can we look at? Well, there's other things you can look at in the body and in the blood, specifically in the plasma. Here, we're looking at something called BDNF. BDNF is brain-derived neurotrophic factor. This is a particular protein in the body, in the brain, that is highly involved in neuroplasticity, highly involved in learning and memory. It's thought to be one of the key mechanisms through which the high doses of psychedelics are able to have therapeutic effects. And even down to the 5 microgram dose, they're seeing significant increases in this LSD microdose. You'll see it's not significant in the 10 microgram condition. You're moving from left to right, 5, 10, and 20. But seeing it in 5 and 20 is telling us that there's something happening here. So don't read too much into the fact that it's not happening at 10. It's just that by chance that didn't become a significant increase, but likely, if they had done the study again, they might see that even with a 10 microgram dose, they're getting these interesting increases in this particular protein in the blood. On the right, on the B panel there, this is just again showing the increase in the plasma levels, blood levels of the LSD at these different doses. So this increase with the different doses happening in association with the BDNF. Okay, so that's just a little bit about what's happening in terms of the blood. It's coming in your body, and it's acting on some of these interesting proteins that might be mediating some of the therapeutic effects. But what about? Let's just jump jump right into the clinical trial here in terms of what people are reporting as far as clinical types of effects, not just from looking at enzymes or proteins in the blood. There's only been one clinical trial so far looking at microdosing, which you can consider a protocol of microdoses across, let's say, a month period, like a Fadiman-style protocol. This actually used a Fadiman-style protocol, so 1 day on, 2 days off, over 6 weeks. So it was a 6-week trial. This could not be done in the United States. The FDA will not allow you to take a microdose home. You have to be under supervision for the entire period that you're on a psychedelic, regardless of the dose. No one's going to be able to provide that much time in their life to sit in a laboratory for 6 to 8 hours a day, every 3 days for 6 weeks. It's just not feasible. But in New Zealand, they prescribe the LSD, they go home and they take it like they're taking any other medication. And this is what this particular clinical trial was. Now, it's the first time they've done this. It was only in healthy men. So we need to have more diversity and understanding how a clinical population might respond. But I'm not going to present these data, but I'll just mention that the findings overall was that with a placebo control, on the days that they took a microdose relative to placebo, they had more increased connectivity reported, more creativity reported, increased energy reported. Also, they didn't expect to have any of these effects. They did a lot of good controls in the study, asking them, what do you expect these microdoses might do to you? And the people in the study didn't have a lot of experience with psychedelics, let alone microdosing. They had no expectation they would have this type of increased creativity or increased connectivity with other people, which they were reporting relative to placebo. So that was really interesting. But if you looked from before the trial to 6 weeks later, once the trial ended, they looked at whether there would be differences in terms of cognitive or emotional effects, different types of tasks, different types of questionnaires. And they didn't see any changes from before to after the 6 weeks period. But those days they took microdoses, they did see changes. So if people can get relief when they're taking a substance, then maybe that's great. One thing they also found was that the day after they took the microdose, that night they slept better than the placebo condition. So they had these wristbands that could track sleep. So that was something that was also really interesting too, that came out of that. Again, they're all healthy people. So maybe there's this thing called a ceiling effect where once you're, if you're already healthy and we give you a medication, are you going to get much healthier? Maybe not as much as someone who really needs the medication and could benefit from that. So there's follow-up studies that are on the way. So anyway, that's kind of jumping from, the more of the kind of basic pharmacology into what do we actually know about microdoses in a clinical trial. The rest of this talk is going to be about the neuroscience, what's happening in the brain. And I just want to point out that a lot of the neuroscience in terms of higher doses of psychedelics is fairly well understood in the last few years. There have been some really interesting papers that have come out that are even superseding what has been written in these particular review papers. But yeah, if you'd like to take a photo, these are the key review papers that have come out in the last few years. But as I mentioned, they're also somewhat outdated. So this is one paper that came out this year that was really impactful. This was in Nature. This is where we were looking at how these substances are acting on the individual neuron level, and this interesting effect known as psychoplastogenicity. So that's, I think, the most psychedelic word that you'll probably hear. But basically, it's this term that was coined to talk about the increase in the connections that neurons make to one another. And this little cartoon shows a really great example of that. You've heard of neuroplasticity before. This is the same idea. It's this neuroplasticity where this psychedelic, they call it a psychoplastogen, is able to induce these changes, like in this case, dendritic arborization, where the dendrites of the neurons are much more varied and able to, you can imagine, connect more easily or potentially restore certain types of connections and activity that had been lost, such as from some type of trauma that they would induce in some of these animals, for instance, in these studies. And so that's what they're showing here in the case of DMT, dimethyltryptamine, which is one of these psychedelics that acts on the 5-HT2A receptor. What this particular study found was that these psychedelics are unique in their ability to actually permeate that membrane of these neurons and get inside and act on all these 5-HT2A receptors inside the neuron, and that allows for this interesting psychoplastogenicity effect to happen, which doesn't happen naturally in the brain when serotonin is around, because serotonin can't get inside the neurons like these psychedelics can. And so this is something unique, it seems, to psychedelics to do this. So that's what's happening on this like individual neuron level. What about what happens in more of like a brain-wide level, like a network level? Well, you have these 5-HT2A receptors. This is a serotonin receptor, by the way, that the psychedelics act on to mediate their effects. You have them not only inside the neuron, but you also have them in important places on the neuron itself, such as these axon terminals that I'm pointing out here. This is where the neuron's actually going to send a signal out to another neuron to communicate. So the axon terminal is allowing the neuron to send a neurotransmitter to the next neuron in the chain to send a signal. What happens when the 5-HT2A receptor is activated by, let's say, psilocybin? You get more neurotransmission out of that axon terminal. And what happens is you have more connectivity, in a sense. In this particular image, you might have seen before, it's from Robin Cart-Harris's group, Placebo on the left. What they're showing is a neuroimaging study. The different colors, the different nodes on the ring are different brain regions. Placebo is how they're normally communicating with one another. The psilocybin condition is showing how there is this hyper connectivity happening, more communication across the brain. When there's more communication brain-wide, the communication that usually happens in some of these modular regions, you might have heard of the default mode network, for instance, is going down. So it's kind of giving itself up to the whole, so to say. The default mode network, what that is the network that's active when you're just resting, when you're just sitting there with your thoughts silently. You might lead to certain places about yourself and your worries and your ego and your things that you got to deal with, and that's hyperactive in depression. Here, you have this ego dissolution experience, which correlates with what they call disintegration of this default mode network, and you're able to basically give yourself up to this larger whole, and that is associated with clinical outcomes in depression. Okay, this also goes back to this major theory for how psychedelics act, which is called the entropic brain hypothesis. Has anyone heard of this entropic brain theory? Okay, so the idea here is very similar to what we're seeing here. By the way, these are these fibers that project out from this region called the thalamus. You can kind of just visualize here all these different axons and where they're terminating and sending out these glutamate neurotransmitters to act on different regions of the brain. Anyway, okay. So, but if you look at this image of the psilocybin on the right, you might think initially it's pretty chaotic looking. You know, there's a lot of another word, entropy happening here. There's a lot of things happening here. So this has led partly to this idea of this entropic brain hypothesis, which is that you might have heard this analogy too. You maybe in your anxieties and your depressions, you have this very rigid structure of the brain, as if it's like a sled that has kept on, going down the same path and is very fixed. And then now imagine a new fallen snow has come, or another snow analogy, shaking the snow globe and allowing all of that to reset. So these are kind of common analogies you'll hear in popular psychedelic neuroscience. This is what the entropic brain hypothesis is really talking about. It's this introduction of entropy, which might relate to the richness of the experience and also the ability to reset some of these connections, and also allowing you to shake some of what they called like the priors, those prior experiences, those prior expectations, and seeing things in a more refreshed type of way. So this is all some of the psychology that's associated with this increase in complexity in the brain signal, related again to these increased connections, increased neurotransmission that's happening from psychedelics. So we see increases in complexity outside of psychedelics. We see there's this range of complexity, and it's used as a neural correlate of consciousness. So as I mentioned in the beginning, let's say you're someone who's in coma. You're going to have very low levels of complexity in your brain, which makes sense, right? If you think of your brain as a machine, let's say, it probably doesn't need to be super complex and active when you're in coma. You're trying to maybe get it out of that state. Anesthesia is another one. So anesthesia, also very low complexity. Sleep, low complexity, but it keeps going higher and higher when you come from these states into wakeful states. There's a certain amount of complexity that all of us have now. And if you're drowsy, then you have a little less complexity. But if you're kind of awake more, then you have more complexity. And if you're on a psychedelic substance, you have even more complexity. So why is this? Why is this that we're seeing this increased complexity? Is it related exactly to the altered state effects, or could we have it without the altered state effects? Is it something that could happen just from the fact there's a little more glutamate firing off, that there's more connectivity? So my question was, can we get more complexity, more consciousness from even a microdose? Because right now the field was saying, okay, this increase in complexity is related to the altered state of consciousness, that richness of experience idea. They're thinking that, okay, all this wild stuff that we're seeing, maybe that's just this increased complexity that's happening, but maybe not. Maybe we can get increased complexity even from a microdose, and this is more related to just the basic physiology and pharmacology of the substance, not so much these intense altered state experiences, which may be related to other types of brain markers, not just this increase in complexity. It also doesn't quite make sense for there to be, let's say, this complexity is a consciousness measure. If you're having a really intense psychedelic experience, you might not be as conscious in some ways. You might be kind of intoxicated, kind of impaired. So it wouldn't really quite make sense that you would have this be a marker of altered states of consciousness if it's really just a marker of this linear increase in consciousness. I'll get back to that a little bit later. It's something to kind of keep in mind. Okay, so what do we see in the brain in terms of microdosing? There's been a couple different studies that have come out, and then I'll show you some unpublished data. This was the first one that we did at the University of Chicago with a technique called fMRI. With fMRI, you can see where in the brain there is activation. In this particular case, they're just having someone go into these fMRI scanners, and they just sit there silently. They're not doing anything. It's called resting state. What they found was that there was an increase in connection, which is basically just two regions of the brain being active at the same time, increase in their connectivity in two important brain regions. One is the prefrontal cortex, which is where you have cognition, executive function, and another one is the amygdala, which is where you have emotional responses like fear responses. And that's what they're showing on this bottom line, that x-axis there. You can see amygdala to MFG is medial frontal gyrus connectivity. So there's increased connectivity. You can think about this as being the logical part of your brain, that frontal executive control part of your brain, controlling the emotional reactivity part of your brain. And so when you have this increased connectivity, you usually see that with someone's ability to have some cognitive control, top-down control is what they refer to it, top being higher regions of the brain, down on the amygdala, control over your emotional states. They saw that the more connected these brain regions were, the more top-down control they had, the more positive mood the participants were reporting. So you can see that on the y-axis there, the positive mood, LSD minus placebo. These are the participants in the study. Each point on this graph is a participant, each one showing this correlation between this increase in positive mood and this increase in connectivity of these regions. To me, this is kind of like the microdose saying, I've got this under control. Like everything's gonna be okay. It's that logical part of your brain is intact and it's highly functioning, and sending those top-down signals down to the amygdala, telling the amygdala, chill out, I got this, everything's fine. If you don't have that frontal part of your brain intact, then you have this disinhibited amygdala that's running around, screaming or something like that, I imagine. So this is my interpretation of these data, and why I think it's important to talk about here. There are other ways of looking at the brain, not just finding out where there are certain brain responses happening, but more like when. And by when, I mean looking at brain waves, looking at brain responses like reflex in the brain. So when someone hits your knee, you'll have a reflex with your foot. You'll have that with the brain. If someone shines a light in your eye, your brain responds in a certain way. And we can do different tasks where we can look how the brain's responding to emotional faces or how the brain's responding to earning rewards. And we can see how microdoses of LSD might affect that while also affecting the resting brain, just looking at the brain at rest and listening into the brain waves. And I'll talk a little bit about brain waves and ultimately, finally, the complexity of the brain. And so with this technique, we use EEG. So it's not fMRI, it's this other type of imaging technology. So you can see it on the right there, this participant just sitting in. This is the study that I was working on when I joined at the University of Chicago. And this is the session room that we use. I went to Indiana University, so I put my undergrad flag up there. I kind of designed this. I brought in my guitar case and chessboard and wanted to make it like a comfortable environment for the participants. So we gave them doses of either placebo, 13 micrograms, or 26 micrograms of LSD, which means that it was a study where they came for 3 laboratory visits. On any visit, they didn't know what they were going to receive. Again, they didn't know they'd receive a microdose of LSD at all. They could have received a stimulant or a sedative or cannabinoid. It was all these drugs that are listed on the consent form. They came in, they're like, all right, let's go. I guess this is the drug lab. And then we gave them a little bit of microdose of LSD or placebo. And then in the middle of the day, they would get their brain recorded. And so this is just giving you a sense of how those microdoses felt for them. What I'm showing you here on the left is you can kind of see what we're used to, this kind of session of, in this case, 5 hours here, 4.5, 5 hours ultimately. And then on the y-axis here is their rating on a scale of 0 to 100. Each time I come in and check in on you, how much do you feel a drug effect right now? So the baseline, everyone, of course, is just zeroed out. But you can see we do the EEG right around peak effects. But the really important thing I want to show you here is that the lighter gray is placebo. The little bit darker gray is the 13 microgram dose, essentially indistinguishable from placebo. So this lower dose of LSD we gave them is right about what they're reporting as a placebo effect. So they don't know whether they're on a microdose at all. If we give them the larger microdose, the 26 microgram dose, this might be what we'd refer to as the museum dose, as I heard earlier today, or other people have referred to as the threshold dose. So they've kind of broken through this threshold. They can tell now here what I'm showing you is statistically significant difference from placebo. They know they're on some sort of drug, but that's only what we've asked them so far here is, do you feel a drug? Of course, that's a very blunt measure. We want to know a little bit more about what the drug feels like. And so we ask them other types of questions like, is it LSD-like? Is it stimulant-like? We don't just ask them that straight out. There's actually a whole questionnaire, and we compile up all of those individual questions together and average them out. And then that gives us this LSD-like scale or the stimulant-like scale. And what I want to point out here is that even at that 13 microgram dose, even though they didn't really feel any different in terms of an overall drug effect, when we asked them questions related to how stimulated they feel that particular time, the 13 microgram dose actually did significantly increase relative to placebo how stimulated they were on that particular time point on that particular day. And this goes back to the pharmacology kind of questions come up here is like LSD versus psilocybin. LSD acts on the dopamine system in ways that psilocybin won't. Dopamine is what is being acted on when someone takes cocaine or methamphetamine, those stimulant drugs. So it makes sense that you might see some stimulant-like responses with LSD here. Later on in the talk, I'll compare methamphetamine to this LSD group, which is important because if the things that we see in the brain that are happening from microdoses of LSD are also happening from methamphetamine, then it's not really that interesting. This isn't really special anymore. This is just maybe a stimulant-like effect in the brain. So we'll kind of keep that in mind for later on. Mood effects. People reported increase in positive mood, increases in elation. And the same people also reported increases in anxiety at the same time. So I think there's kind of this increase of just sensitivity. I feel elated, but also a little, sometimes people will be kind of doing this with their fingers a little bit, feeling a little nervous. So this is what we're seeing in the placebo-controlled design. I think it's, reinforces what, in a sense, we've already known from the self-report data that James Fadiman has. I just wanted to point this out real quick. We have this really cool altered states of consciousness scale, and it has really fun subscales like oceanic boundlessness, dread of ego dissolution, visionary restructuralization. Dietrich, this guy in Germany back in the 80s, put this together, and Rahul Khan, who's at USC now, actually translated it into English. But this is a way of assessing the psychedelic-like effects of microdoses. So we wanted to make sure that these people weren't having psychedelic-like effects. We wanted to make sure they weren't hallucinating, that our doses were truly, in the sense, microdoses where it's kind of below that threshold. One thing we did see, though, is that there was a significant increase in blissful state. So, it's not a altered state-like effect, but it is on the questionnaire. And so I thought that was nice that they did report increases in blissful state, but they weren't reporting increases in any of this other stuff like experience of unity. By the way, this y-axis in this case, this is 0 to 100 is the top that they could reach here. And so these are all very, very subtle. Even the blissful state, they're only getting, it's like a 1 out of 10 of blissful state, essentially, is this average for the 26 microgram dose. But again, the bottom line here is that they are not having altered states of consciousness from these microdoses. Getting into a little bit about what we looked at in terms of the brain responses, we found two things. One is that the microdoses of LSD do indeed actually modulate brain responses, those reflexes I was talking about, and they also affected these resting state brain waves. And so I'll talk to you a little bit about what exactly we found and why I think it's interesting. There's a lot happening on this particular slide. All I want to show you is that this 13 microgram dose is giving this big response here called the RUP. The RUP is related to your response to rewards. So if I, basically, have you play a game, which is what they're kind of doing here. They're on the computer, they're pressing buttons, and if they win in the game, they actually earn money. The moment that they win, that is the 0 point, which is probably too small to see here, but this line going down, that's 0. So as soon as they see win on the screen, their brain's going to have a response. That response is this RUP section right here that happens about 200 milliseconds after that stimulus goes off. The 13 microgram condition facilitated that brain response. It's like, oh my gosh, I just won. Interestingly, the placebo group was the same as the 26 microgram group, the higher dose in terms of its response to reward. What I'm trying to kind of suggest here is that there might be a sweet spot in terms of this facilitation in a reward-like response. Imagine if you took a very large macrodose and you're trying to play this game and you won. Would you really be tuned in to winning at that time? Probably not. So it might actually go down relative to placebo. Whereas if you're just maybe at these kind of sweet spot doses, you're tuned in, you're attentive, and you're able to receive this type of reward in a way that you wouldn't normally be able to do on those higher doses. So that's a finding that I'd love to follow up on later to see if that's something that we can validate later on. There were also responses to faces. I don't have those data that I'm presenting here, but the bottom line with that was that the brain responses to angry faces was lessened. So they didn't have this sort of maybe emotional response to the angry face, but they actually were better at button pressing for distinguishing angry faces from neutral faces from happy faces. So this actually improved their behavior, these microdoses, in addition to affecting their brain responses. Okay, so let's talk a little bit about brain waves and try to break it down for you a little bit here. So you probably heard about, brain waves with meditation and things like that. And so one question is, does a psychedelic put you more into a theta state or an alpha state? One really interesting thing happens actually with the large doses of psychedelics, which is that it takes all those brain waves from delta through gamma and it desynchronizes them, which means that it reduces them. And it's this really interesting effect. And there was a paper that came out in 2013 that had this big fancy title, Broadband Cortical Desynchronization Underlies the Human Psychedelic State. What does that mean? It just means brain waves are lessened across the cortex, across the brain. Whether you're looking at delta, whether you're looking at gamma, whatever it was, those amplitudes of those brain waves get flattened. Why does that happen? Well, when you're in a rested state, let's say you're sleeping, for instance, you might have heard you have those really big delta waves because the brain isn't really doing anything at that time. You're not having to worry about thinking about too much. You're just letting your brain rest. Of course, your brain's still alive. And so what happens is your brain is oscillating in this kind of rested hibernation almost sort of mode. This is actually something that happens in nature generally. So there's really interesting examples of this where systems will tend to oscillate with one another when they're in a state of rest. So there's, as an example, they'll have like a table with metronomes, and they start these analog metronomes at different times, and they're all clicking at different times. But because the table is able to kind of vibe with all the metronomes in a sense, communicating, all of a sudden they'll start to, over time, now all the metronomes are in sync with one another until you disrupt the table again, or something like that. So as long as the system, and I kind of think about this with people, if you have enough people in the room, maybe heartbeats will sort of beat in the same rates if they're sitting there, things like that. You can kind of imagine different cases where these systems will oscillate in sync. That's what happens with the brain. That's where you get these big delta waves that come from. Another example of this is with alpha waves. So as you move from the low frequency delta to the higher frequencies like gamma, you can think about it as like a little bit more of an active type of state. So when right now we don't have delta happening, but we might be in some theta alpha is the next level. Alpha, and a really good example of alpha is if you were to close your eyes, you don't have to close your eyes, but if you imagine closing your eyes right now, what immediately happens is you have a lot of alpha in the occipital lobe. And you're going to see that very clearly. And this is when, it's called alpha because it was the most prominent wave, it was the first one discovered. And that's because the reason why there's a lot of alpha when your eyes are closed is because you're basically resting your occipital lobe back there, the lobe of your brain responsible for vision. The neurons don't have to do anything. They're just kind of vibing. They're just in this rested state. And when you open your eyes, the wave immediately desynchronizes. It reduces because now these individual neurons, as opposed to communicating in this synchronization with one another, are now having to individually figure out shapes and colors and this and that, communicate to different regions of the brain. And you get this reduction in those amplitudes because you've made the brain more active. Looking at psilocybin, that's basically what we can glean from this. We have reductions in delta, reductions in alpha, reductions in all these brain waves. Broadband means from delta through gamma. Band is referring to this band of frequency, the delta band, the theta band. Broadband is all these frequency bands are desynchronized. You can imagine because the brain has become more active, that more glutamate transmission, more interconnectedness. So that was one thing we wanted to look at with microdosing. Does that also happen with microdoses? Does it truly underlie the psychedelic states? Is it related to the experience of psychedelics, or is it just something that happens even from a microdose because of the pharmacology, something you can actually have without having those altered state effects? This goes back to this entropic brain idea as well, which is, can we also have this increase in entropy, this increase in this complexity measure, this increase in this consciousness measure? To answer the question, yes. I'm not showing the data here, but this paper that I published in 2021 found that we also got these these desynchronizations in the brain waves, even from a microdose. I'll kind of show you the new data that adds more color to that. This was one example where the answer to the question, do microdoses reproduce the effects that we see from macrodoses, was yes. There was this cortical desynchronization that happened even from a microdose, and it's not something that's specific to a psychedelic state or altered state. It's just even a microdose can have these interesting brain effects that may be therapeutic related. Maybe we don't really necessarily need to have those altered state experiences, even if they can be beneficial, and add another layer to the therapy. Maybe the bulk of the therapy can even happen at some of these lower doses. Okay. So the entropic brain, that was the next question was, okay, we saw that we had interesting brain modulations to those responses. We saw that the microdoses were able to reduce those brain waves. What about increasing entropy in the brain? What about this complexity consciousness measure? So the unpublished data, I'm pulling the data from the microdosing LSD study, but now I'm comparing it to a THC study and a methamphetamine study. These are really important comparators, different conditions to demonstrate whether or not the LSD, the psychedelic, is special in a sense. If we see the same type of brain responses from methamphetamine, then it's not really a psychedelic thing we're looking at. If we see the same type of things happening in THC, this is important because these THC doses actually induced altered states of consciousness, unlike the microdoses of LSD. I'm going to say that one more time just to kind of drive the point home of why we're looking at THC here, is because the THC was psychedelic. The THC had that psychedelic effect, that adjective psychedelic. It was inducing really intense altered states experiences. People were having very interesting types of psychedelic responses. And so if we see the same type of entropic brain under the THC condition, then maybe we can say, okay, this must be related to psychedelic experiences. But if we don't see entropic brain under the THC condition, then it must not be really related to psychedelic experiences. It must be related to the pharmacology, the psychedelic noun, as opposed to the psychedelic adjective, which the LSD microdoses are that psychedelic noun. What we're looking at here is Limpel-Ziv complexity. So this is this very common measure of neural entropy, very common measure used in these coma studies, anesthesia studies. And so here we're going to use it for microdoses and then compare it to these other doses. All right, so this particular slide is just showing us these overall subjective effects, how much people feel it. You've already seen the LSD condition here, how the placebo dose was essentially the same as the 13 microgram dose and the 26 micrograms was, noticeable. How did that compare to these other doses that people are getting with THC and methamphetamine? So here you can kind of see the 7.5 and 15 milligram conditions mapped onto each other pretty well. This is orally administered, so we gave them a pill. So 15 milligrams is essentially like giving someone 3 standard edible doses. These people were very intoxicated from cannabis. I checked in on one person, and he was on the couch laughing, trying to explain what it was he was experiencing, and sometimes people would just fall asleep, and they would be out. And I wasn't there for the methamphetamine study, so I don't know how those participants responded to these doses, but it gives you some insight into how it compares across these 3 groups. So these are 3 groups of people, but every group, they came for 3 different sessions to receive placebo or these other 2 doses. And importantly, all of these people aren't heavy substance users. The people who are in the methamphetamine condition aren't methamphetamine users. The people in the LSD condition, they're not LSD users. They might have used it once or twice. And again, the people in the THC condition, they're not THC users, not heavy cannabis using folks. They're people who have experienced it actually in this study less than 20 times. So similar to the amounts of methamphetamine use or the amounts of LSD use in these other folks. So that's one way of kind of having some similarities that we can try to draw from, and they were similar ages. They're all about 25 years old, and average about from like 21 to 30 something. So similar groups of people across these 3 different studies. What are their mood effects like? I already showed with the LSD, they had anxiety and elation at the same time. Well, they really had anxiety from THC. This particular group, they didn't really have experience with cannabis before, and they really didn't like it. They weren't always feeling super good. In fact, their elation went down, relative to placebo a bit. Methamphetamine, people felt pretty good from that. They were going up, a little bit of anxiety. So this just gives you a sense of, okay, we are getting drug effects. We are getting these stimulant-like effects, of course, happening with methamphetamine. And the mood effects, I think, are important when we're trying to figure out what we're looking at in the brain. Is it tied to a certain mood-related effect? The altered states of consciousness, we didn't administer this to the methamphetamine group. If I was at the University of Chicago still when they did it, I would have had them do it. But we can assume that they weren't having altered states of consciousness from methamphetamine. They really weren't having it at all from the LSD. Again, this is a 0 to 100 scale. You can see it's right all around 0 here. All of these different subscales of altered states, nothing happening. Whereas THC, at every single one of these is highly significant increases in auditory alterations, in oceanic boundlessness. This includes things like insightfulness. Insightfulness is one of the subscales of these as well. So there's a lot of things going on in the minds of the people who are taking THC or cannabis, especially if it's one of the first times they've had it in a very high dose. In some cases that may be paranoia. Some cases it may be more of euphoria. So a lot of things are going on that just aren't happening with the LSD. So THC in this case is psychedelic. LSD in this case is not psychedelic. LSD is the psychedelic noun, but THC is the psychedelic adjective here. All right, let's cut to the chase and look at the measure of consciousness, this neural complexity measure. So here what I'm showing are different electrodes on the brain, on the scalp rather. And it's kind of hard to tell here, but this is a nose at the top and you got ears on the side. So this is looking at the head from above. And each one of these circles is another electrode on that head cap. And what I'm showing you here is the 3 drug conditions, LSD, THC, and methamphetamine. And on the left-hand side is the lower of the 2 doses. On the right-hand side is the higher of the 2 doses. And what I'm showing on the right next to that is this color spectrum relative to placebo. So placebo would be 0%, you could say. The change from placebo is 0, which would be white. So in this case, like let's say this little electrode here, when I look at that, relative to placebo, this electrode really didn't change much in terms of this neural complexity. But in the cases where it's very bright red, dark red, there were big increases in that complexity measure. Cases where it was blue, there were decreases in that complexity measure. If it has a yellow circle around it, that means it was a significant increase in complexity after correcting for multiple comparisons. So statistically significant increase at that particular site on the scalp, giving us some insight into the spatial resolution, even though typically this type of imaging technology is not considered to be very good at spatial localization. It surprised me to see the type of pattern that emerged here, which was somewhat lateralized. I can go into more detail about that later, but the point to bring across was that only the LSD condition, not the psychedelic THC, not the stimulating methamphetamine, only the microdosing LSD condition is having these significant increases in this complexity measure. On the right here, I'm showing bar graphs that are just essentially averaging all of the electrodes together. And we can see that there's this significant increase relative to placebo at this 26 microgram dose. But even at that lower dose, that 13 microgram dose, where there was essentially, it was indistinguishable from placebo and an overall field drug effect, there's still, a lot of, dark red coming out. So really interesting to see that, even at these lower doses, we're getting this increase, this entropic brain, this increase in neural complexity, this marker of consciousness, which we don't see from THC or methamphetamine doses, even when they're pretty high doses. I mentioned earlier that I was going to add more color to those brain waves I was talking about. So this is really getting into the weeds a little bit, but if folks have certain interests in how microdoses of LSD affect delta or theta, I can kind of touch on that right now. We refer to this as spectral power. So you can think of the spectrum from delta through gamma. Power is referring to the amplitudes of those brain waves. So if you have a high alpha power, that's what happens when you close your eyes. You have high alpha power closing your eyes, lower alpha power when you open your eyes. Here, what we see is that the microdoses of LSD lower the power of delta and theta. So big blue here, blue is lower, less relative to placebo. So they're reducing delta, they're reducing theta, where we're not seeing that with THC or methamphetamine conditions so much. They're also increasing gamma a little bit. So as I mentioned before, the initial study I published was that all the brain waves from delta through gamma were having this reduction. We didn't really quite see that when I did the reanalysis here, because in the previous analysis, we just focused on the default mode network regions, which are really in the middle, this midline section of the brain. And if I look at gamma here, for instance, you can see there's blue decreases in that midline section. So if we just focus on the midline and these kind of more default mode network regions, we would see these desynchronizations. But when we look at the whole brain, we see kind of more of a nuanced pattern where it's really only delta and theta where we're seeing like these brain-wide desynchronizations in those bands, and maybe even some increases in gamma. The final thing I wanted to point out here is not related to the microdoses of LSD, but comparisons, an interesting contrast between the THC and methamphetamine condition. It's specifically at alpha. So as I mentioned, alpha, when you close your eyes, you have big alpha happening in the back of the brain. If you can look closely, you can see the methamphetamine condition had increases in alpha in the front of the brain, as if the front of the brain was kind of quiet. Maybe it also means it was more efficient. They're just sitting there in this state of mind that nothing's really happening. If they have a thought, it might come and go quite easily. This is opposite to the THC condition. The THC condition had big reductions in alpha, something we didn't see in the LSD state. Big reductions in alpha mean that the brain is more active, essentially. Maybe there's more processing of paranoia, processing all of those altered states experiences, insightfulness, oceanic boundlessness that might have been happening that we just don't see in the microdosing condition. So for me, and this is actually something I looked in the literature a little bit more, in addition to these ideas of maybe neural complexity being related to the psychedelic state or decreases in broadband power, alpha power in particular has been another one that might be this main signature of these altered states. And that's something I'd like to look at a little bit more closely. These decreases in alpha may be directly linked to altered states of consciousness, whereas increases in complexity may be more related to just this level of consciousness without the altered states, just enhanced ability maybe to have improved therapeutic outcomes, improved behavioral outcomes. We don't know. That's where this study stops short, is I have no idea what the benefit, if any, is of this increased complexity from the microdoses, but the future studies should really dig in closely. What is the clinical, cognitive, behavioral relevance of the increased complexity from microdoses? And also, what is the functional significance of this desynchronization, reduction in alpha from THC? Is it truly reflective of these altered states of consciousness? A couple last slides here. This is just showing a big correlation plot, just trying to get a sense of connecting the brain to the mind. What is it that we see in terms of, saw those reductions in delta and theta? Are they related to anything with how people feel the drug, anxiety, elation? There's really nothing I really want to point out here. This wasn't really a super interesting part of the paper that I'm putting forward, other than to say that there were some associations between those decreases in delta and theta from LSD and their elation responses. So that decrease in delta and theta that we were talking about earlier with LSD could be related to the improved mood effect to some degree. So we're just kind of checking for correlations here. Nothing too crazy or anything I want to drive home too much about. This is interesting though. So this is, before I conclude, this is one kind of fun thing we were able to do in the study, which is, okay, you can look at these interesting brain measures like complexity, and you can look at them with respect to a drug intervention. But what about other variables aside from a drug? What about things like age? What about things like sex? How do other individual differences in people affect these different brain measures? And so what I did here is I'm looking at delta, theta, alpha, beta, gamma in this complexity measure in two different age groups. We have 18 to 20-year-olds, and we have 30 to 40-year-olds. And what I'm showing here are the adult group relative to that adolescent group. So if we have decreases here in delta, these are adults have less delta power than adolescents. As the brain gets more mature, these adults, you kind of think of this as their peak development period, whereas the adolescents are still fledglings. They're still kind of getting up. They're not quite fully developed yet, right? These folks in their more developed state are going to have less delta, less theta than the adolescents. There's a little bit of significant increases here happening. Not a lot of happening in alpha or beta, and some increases in complexity. Now, if you've been awake enough to remember all of the previous slides I've been showing you, you might think, hey, isn't that what LSD did? Isn't that exactly the same pattern of the microdoses of LSD? The microdoses of LSD reduced delta, they reduced theta. There wasn't really anything happening with alpha. It was meth that increased alpha and THC decreased it. And the microdoses of LSD also, they kind of increased gamma a little bit. It wasn't significant, but they definitely increased complexity. And so I think this is a really interesting thing where are the microdoses of LSD replicating this brain maturation state? And kind of one hot take, which I don't want to try to sell too hard, is that when we think about the methamphetamine condition, if this is really similar to Adderall, and we're giving children Adderall and their brain becomes kind of the zombie state where there's just nothing going on, they're dead silent, they can take information in and spit it back out, but maybe it's healthier potentially to replicate an adult's brain than replicate this thing that doesn't happen only with these types of stimulant drugs. So that's kind of a hot take, but I think it's interesting that the LSD effects that we're looking at and are the same pattern as we see with a healthy developing brain. Okay, so in conclusion, the very low doses of LSD, which are not readily distinguished from placebo, are nonetheless sufficient to reproduce these effects that we see from the larger doses, such as psychedelic-assisted psychotherapy. I'm not saying it's the same magnitude of effect. You might have much larger magnitude of effects from the psychedelic-assisted psychotherapy. You can kind of imagine this as like the dials turned down a little bit, but it's still significantly higher than placebo. So it's the same direction of effect, the effects happening, but probably not the same magnitude. It'd be interesting to have a head-to-head study to see just how much that dial's turned down. These may be therapeutic effects, and they seem to be largely unique to the 5-HT2A agonists. I want to say that we do see this with ketamine. So I've been talking about the specialness of psychedelics. The pharmacology after the 5-HT2A receptor, what's downstream of that receptor, is very overlapping with the pharmacology of ketamine. The literature has reported increased complexity from ketamine as well. So you kind of group ketamine into almost the psychedelic noun in a sense in terms of its pharmacology, even though it's not a drug that is acting on 5-HT2A receptors, it acts on NMDA receptors. So in some cases, though, what's interesting too is that these low doses may enhance brain responses in places that the higher doses cannot. So it's possible that the microdoses can do things that even a high dose can't do, in addition to kind of replicating what the high doses can do. So we mentioned that sweet spot dose where maybe we can be more receptive, rewards, for instance, and being able to respond to different stimuli. This idea that the altered states of consciousness are not linked to the increase in complexity, but may be inherently linked to alpha desynchronizations is another finding here I'd like to follow up on. And so characterizing the role of alpha desynchronization for consciousness and cognition, what is it really doing in terms of these altered states? And again, finally, the big missing piece, the big gap in knowledge is what is the clinical, behavioral, and cognitive relevance of this greater complexity after low doses of LSD? Is it actually helpful? Maybe it's just an increase in noise in the brain? So that's one thing I didn't mention when introducing this idea of complexity in the brain, was that traditionally, historically, folks thought of complexity as just background noise. And maybe having more complexity is actually not a good thing. And so, for instance, there are a couple studies where they found that people with schizophrenia have a little bit more complexity. However, if you look at other brain disorders, it usually almost always reduces complexity. And even in the case of schizophrenia, if you look at the natural increase in complexity that happens with age, like how I independently reported here, people with schizophrenia will have less complexity in terms of the age-related increase. So schizophrenia is blunting that neurodevelopmental increase in complexity in another paper. So the data on schizophrenia is mixed in terms of complexity, but in general, having more complexity is a good thing. And when you have less complexity, it's considered like a disorder of consciousness in some sense. So what is the relevance here? Is it actually doing something from microdoses of LSD? Are they getting better? Is it related to behavioral improvements? Is it related to clinical outcomes? All right, so that is the presentation. I'd like to acknowledge the University of Chicago where this work was done, the Human Behavioral Pharmacology Lab with Harriet de Wit and all those folks, and as well District216. So thanks for having me.
Are there data from other studies on psilocybin that would allow you to, in general, make a comparison about brain complexity, comparison with LSD and psilocybin?
Yeah, so there definitely is in the larger doses. This entropic brain hypothesis idea, this increasing complexity is from the large doses of psilocybin, but no one's done a microdose psilocybin study yet. But I would, I'd be surprised, I'd be shocked if they wouldn't be able to find that. There was one study that did a little bit. It was kind of like a take-home study with psilocybin, and they looked for entropy and they didn't find it. But it wasn't like as controlled of a study as this one. So I would assume if I replicated all of this with microdoses of psilocybin that we would see it, but the data's not out yet.
And I was surprised to see that the low dose of LSD didn't have much effect. I thought it should have more effect. It has more effect on me, I'll say that.
Yeah, so, it's hard, too, to really figure out the dosing because it's hard to get these doses into a drug solution, and that's kind of a challenge. So it's possible that some of the, like if you were taking like a 10 microgram dose or a 20 microgram dose, it might be a little bit different than 1 microgram dose.
You were talking about doing cannabis in your youth, and I heard that it's not good for young people while their brains are developing. Any thoughts about that?
Oh yeah, absolutely. I was trying to think when's the best time of your life to ever do cannabis, and I came out to the conclusion that really never. But sorry, but anyway, yeah. So yes. There are a lot of studies that show that the brain's still developing, and that if you, one researcher referred to as throwing a wrench in the system of a developing brain, then you might cause some things down the road to not be so great. But there's a lot of one. I don't have the data here in front of me, but one thing I found that was really interesting that I can speak to in terms of a finding that's not yet published was that we use these EEG headbands on 100 people that came into the laboratory. I currently work in the Center for Cannabis and Cannabinoids at UCLA. These people ranged from 21 to 55 years of age, and they had a range of using cannabis on average from 0 days of the year to 365 days out of the year, so daily. And they were men and women. And what we looked at was that a marker of cognition, or a marker of brain development. I kind of mentioned earlier the delta and theta, how we saw those decreases in terms of folks who are a little bit more mature. And so essentially we were looking at that delta and theta and the other brain waves, and in the frontal lobe where we would expect to find this adult-looking brain. And so the question was, and this is from preclinical literature, the preclinical literature shows that if you give really high doses of cannabinoids to young rats, and then you look at their brains as adults, their brains still look like young rats. And so the question was, does that happen in humans? Can we try to replicate that in humans? So I have this brain marker of adulthood, basically, and I wanted to see, do the adult brains look like adult brains, or the more cannabis they're using, do they look more like an adolescent brain? And what we found was that particularly in young men, so the college-age guys that came in, the more cannabis they were using out of the year, the more their brain looks like a teenage guy brain, basically. The women, though, were protected from this. They didn't have that, which was interesting. So those are my data. It will be kind of controversial, but overall, when we had all the participants together, and Steve Grant, will could poke a hole in this, because the more people you have in the data set, the more likely you are to get a significant effect. But when I parsed it apart, and I found that effect was driven really by this one small group of these young men, these 21 to 23-year-old men in the data set. And so I am kind of concerned about that type of issue, and maybe the women were protected because their brains develop more quickly, and so the folks who are using cannabis while their brain's still in the midst of its development, might be slowing that developmental process down, and that might result in the preclinical literature. What they say, the concern there is that during your adolescence is the period when you're most vulnerable to psychiatric disorders like psychosis or depression or this or that or the other. If you're extending that adolescent brain state, then you're extending your window of introducing psychiatric disorders, which is where they might be seeing these correlations. There's a lot of correlations with heavy cannabis use, frequent cannabis use, and high dose cannabis use with psychiatric disorders like psychosis. And I think when I talk to people who are in the medical field and they actually work with folks that get introduced to the emergency department, it'll be young men who are also, coming to the emergency department as well. So so young guys, I think it's important for those data to be out. So I appreciate you asking that question.
But you worked out.
Well, so I wasn't using frequently. I was very intentionally trying to have the lowest tolerance as possible so I could enter these states. And so you can almost imagine, using cannabis recreationally would have been like sacrilegious in that type of sense, even though I did, break my own rule, of course, in different instances. But so I never was a heavy user. But it's important that, I think mental hygiene for young people, I think is important. That's a good question. So if I looked at the older group, eventually it looked like they caught up, so it's like ultimately your brain can, kind of catch up. And if you get through your adolescent period without developing, a psychiatric disorder, then you're fine. But I think it's just you're increasing the chances. You're kind of inviting things to happen during those younger times. It's just you're adding risk factors. And so we live a life, we take risks, but it's important to have the informed decision making from evidence like this. Otherwise, we're kind of just shooting in the dark sometimes.
I'm a Hoosier as well.
Oh, cool.
Bobby Knight, R.I.P.
Yeah.
When you talk about, I believe you said increase in plasma. What do you mean by that?
Sorry, yeah. So plasma, do you know what plasma is by chance?
Well, I was just thinking like blood plasma.
Yeah, blood plasma, yeah. So they were measuring the LSD in blood plasma.
Oh, okay.
So yeah, it's just a really basic thing did we get the LSD in your body?
It's not increasing like your hematocrit. It's not like...
No, yeah. I was just literally looking at the concentration of LSD. And
that'll be a new one.
Right, no, yeah. It was just a very basic study of, it's kind of an obvious thing. Of course, we're going to find, like if we gave you alcohol and we looked at alcohol in your blood, we would find alcohol. But it was just the very first thing you do in a study is to make sure that the participants actually get the drug you're giving to them. Mm-hmm. And so that's what that particular lab did. Yeah.
Gotcha. Regarding, you mentioned ketamine a couple of times. Is that any interest to compare LSD and ketamine, or what would you think would be the difference?
Yeah, it is of interest. There is a paper that came out in I think it might have been 2017, where they did a head-to-head. It was psilocybin, ketamine, and I think LSD maybe. But they looked at this entropy measure, and ketamine was the substance that increased the entropy the most, this complexity the most. Of course, it's hard to have like dose matching. But my impression of ketamine is that it basically is the same pharmacological mechanism. It increases the BDNF that I was talking about. It acts in these receptors that are downstream of the 5-HT2A in similar ways. At the end, it comes down to how long those effects last in people and how meaningful the experiences are. So I'm very interested in ketamine and those types of comparisons, and there should be more work that looks at that. But from my understanding, they're pretty comparable.
So say with the
That's a great question. So yeah, that's kind of a project I've been working on now, is sending those EEG headbands to people and tracking them over time. And so those long-term effects we see from a single dose of a high dose of psilocybin or a high dose of ketamine, does that happen if people take these repeated doses and they stop after a month? Are they still having these enduring, lasting changes? That's an open question. What I've found so far is kind of complicated, but with giving someone a headband and asking them to wear it every day, what we find is that their biomarkers, their brain markers of brain health, you could say, start to go down if they're taking a placebo over the course of that month, because the way I interpret it is they're losing enthusiasm, their attitude. They're like, I don't want to do this anymore. The first day, they're probably like, this is really cool. And so their brains, kind of hooked into that. What we found with the microdosing group is that they actually stayed at this kind of highly enthusiastic state. They didn't ever go down. And then we did like 4 days of we call post tests, and we didn't see like a big drop off, after they stopped microdosing. It stayed, but it'd be great to look at that, weeks later. Very few people had, increases in some of these like experimental biomarkers. Again, what we're looking at are things like that adult looking brain, experimental markers of anxiety and things like that.
Hello. Yes, I had a question about the entropic brain. Mm-hmm. What if it's not entropic? What if it's merely stochasticity? And we're not talking about degeneration of a thought or a pattern, but merely the hypermutation of it. Has there been, Steve, has there been any experimental data at NIH about that?
So I'm looking up the word stochastic here. Randomly determined, having a random probability distribution or pattern that may be analyzed statistically but may not be predicated precisely. What does stochastic mean in your question?
So I've been researching biological relativity. And biological relativity is based mostly upon stochasticity, and weighted stochasticity at that. So, and my question is, could it be not so much a degeneration of the complexity of the brain, but merely the brain coming up with one solution after another?
I love that. Yeah, that's definitely worth thinking about.
It's not falling apart. Entropy means that there's loss of energy, that there's loss of something.
Yeah. Entropy, when they use it, entropy with like physics, is like the loss, like the entropy of the universe. But I think when they are talking about entropy in these cases, they're referring as just like a word they use for complexity. It's kind of synonymous, But I think the stochasticity thing, I think that's cool. So maybe we'll borrow that.
So you use the term oceanic boundless a number of times.
Uh-huh.
I've heard the term before. I'm unfamiliar with its meaning. Could you elaborate?
Yeah, sure. Okay, so oceanic boundlessness is a subscale of this questionnaire, and it's comprised of even further subscales. So specifically, oceanic boundlessness, the term that this guy Dietrich prescribed, this overall scale, is comprised of experiences like experience of unity, spiritual experience, blissful state, and insightfulness. So he just said, I have these 4 subscales, experience of unity, insightfulness, and I'll call them bundled together oceanic boundlessness. So that's what that's where that word is coming from. So similar, dread of ego dissolution is just a bundling of disembodiment, of anxiety, and of impaired control and cognition. So it's just the way that this particular author devised the questionnaire.
So as a neuroscientist, if you were to recommend somebody to take a macrodose of, say, psilocybin or of LSD, what would you advise them to do to recover from that, from the flooding of the neurotransmitters and so forth?
To recover from it, like afterwards?
Yeah.
So we definitely stress integration. So in our studies, we'll have up to 4 hours. I think if people request more time, I think we can probably give it to them, but at least 2 hours of integration. Integration is just after the session. We're definitely going to follow up and call you, first of all, and then make sure that you're okay, and we'll have some questionnaires for that. But then we're going to actually invite you back and sit with the physician and talk about the experiences that you had. And so it is a lot of kind of the psychotherapeutic buffering around that experience to make sure everything's okay. In terms of recovery, though, you might be asking more about like giving a supplement like 5-HTP or something. Yeah, we don't have that written in the protocol at all, and I actually haven't really read anything about people providing supplements following that. But that's a great suggestion. Yeah.
As like a neuroscientist, though.
Yeah, I think it's valid. I mean, I actually read a paper last week because someone asked me about 5-HTP. So 5-HT is serotonin, and P is this precursor. So 5-HTP is the precursor for serotonin. So if you were to take this supplement, which you can buy at Whole Foods, then the idea would be that it could give you more serotonin levels, with this sort of idea that the serotonin would be depleted after one of these experiences. And I thought that was like, I hadn't heard or read about this sort of idea of depletion of serotonin in the literature too much. And I thought, hearing it from friends, it was kind of like hearsay or sort of like a folk myth or something like that. But then I did find a paper, but it was like 2007. It was really old, but it did say in this animal model, like rats, that they were able to restore levels of serotonin after administering MDMA, I believe.
With 5-HTP?
With 5-HTP, yeah. In some sense, it's kind of common sense, taking these types of supplement, vitamin C, for instance, and just getting rest. And so I think that's part of why researchers haven't been too big. But as we get more standardization in our integration practices and sort of like, I guess what you could call like aftercare, I think that'll be developed much more fully. So yeah, I appreciate you mentioning it now, because we'll probably want to ultimately incorporate some of that stuff.
The dopamine, since the LSD and I think psilocybin to increase dopamine, right?
So LSD, unlike psilocybin, it acts on dopamine receptors, and this would mediate some of those stimulant-like effects that you would see from something like a stimulant drug.
Well, I was thinking for Parkinson's sufferers.
Yeah, so in Parkinson's, you have a dysregulation of dopamine, loss of dopamine cells, and there's this a YouTube video that had actually someone smoking cannabis who had tremors, and it all of a sudden now he was like totally fine after smoking. That was sort of one extreme case. They've tried to follow up on that in clinical trials and haven't seen much of a response. But you can kind of imagine that substances that act through dopamine might be able to restore some of that lost dopamine signal. So yeah, sure, maybe there might be better medications than LSD for that. But yeah, it might help a little. I don't know.
Well, thank you guys all for coming tonight. Can we get one more round of applause?
Thank you guys. Yeah, appreciate it.